Howdy, my name is Angelica Ramirez, and today I'll be presenting Group 3's Phage lab reports. Plaque assay is a technique commonly used for the purpose of growing isolated plaques of phage particles within a lawn of bacteria. The combination of bacteriophages and bacteria creates a clear visualization of the effect bacteriophages have on bacteria. The even distribution achieved by creating a bacterial lawn allows for clear observation of bacterial cells that have been infected and lysed by bacteriophages. The amount of PFU units measured describe the concentration of infectious particles present and determine the infectivity titer. Plaque assays and PFU also assist in identifying viruses, determining antiviral susceptibility, and to compare virus-like particles to determine infectivity. For this lab, we used a 10 to the negative 1 dilution of phage, 4 LB plates, 9 microcentrifuge tubes, sterile water, micropipette and tips, 4 large test tubes, E. coli, and the T4 phage. We started by making a serial dilution by labeling 5 microcentrifuge tubes as 10 to the negative 2, 10 to the negative 3, 10 to the negative 4, 10 to the negative 5, and 10 to the negative 6. In each tube, 900 microliters of sterile water were added to each one. Starting from the 10 to the negative 1 tube provided, 100 microliters were transferred into the tube labeled 10 to the negative 2. From this tube, 100 microliters were transferred into tube 10 to the negative 3. This step was repeated until we reached tube 10 to the negative 6, mixing well in between each transfer. Once the 10 to the negative 6 dilution was completed, 100 microliters were removed and discarded so that each microcentrifuge tube contained 900 microliters of solution. We then proceeded to make a whole plate titer from the serial dilution. Each of the 4 LB plates were labeled to match the dilution that would be inoculated with. 4 new microcentrifuge tubes were labeled E to the negative 3, E negative 4, E negative 5, and E negative 6. From the original serial dilution tubes, 100 microliters of each dilution were added to the matching E-labeled tubes. 100 microliters of the E. coli stock culture was then added to each of the E-labeled microcentrifuge tubes. These tubes were vortexed briefly to mix the solution. Once all of the components had been added to the E-labeled microcentrifuge tubes, they were allowed to rest for 10 minutes. Then we proceeded to finalize our lab by preparing the bacterial lawn. Using 4 large test tubes, 5 milliliters of molten agar were added into each one, allowing some time for the agar to cool before adding 200 microliters of the phage/bacteria mix dilution into each one. Once combined with the molten agar, the mixture was poured into the respective LB plates of the dilution. The lawn was allowed to set for 5 to 10 minutes before inverting the plates and sealing them with parafilm and allowing them to be incubated until the following lab time. These were pictures of our 4 LB plates, starting from the left from the negative to the negative 3, 10 to the negative 4, 10 to the negative 5, and 10 to the negative 6. Based on the results gathered in the LB plates, there were too many plaques to be counted. There are some clear zones where bacterial lysis took place. There was also some visible decrease in the plate quantity as the phage was diluted, but there are still too many plaques to be actually counted individually. We made this calculation based on a theoretical number provided by our TA, which were 170 plaques divided by our dilution factor, which was 10 to the negative 4, times the volume of the diluted virus added, which was 0.1 milliliters, which came up to 1.7 times 10 to the negative times 10 to the 7 PFU per milliliters. The result of this phage experiment proved to have an abundance of phages in the sample tested, to the point where counting them individually was not possible. This indicated a very effective and fast bacteriophage replication, which is why the LB plates contained a dense concentration of phages. A possible error that could have happened was an insufficient dilution during plating that could have accounted for an abundance of plaques on the plates. This concludes our presentation. Thank you.